Hi, my name is Malde and I'm a product manager here at Symcel. Today I would like to introduce to you calScreener Plus biocalorimeter. First, a little bit about Symcel. We are global leaders in isothermal microcalorimetry. Our headquarters is located in Karolinska Institute campus in Stockholm, Sweden. We have offices in Denmark and USA and we are globally available. We consist of more than 45 experts from 18 nationalities. We have a strong scientific foundation when it comes to calorimetry and biology. We have 5 approved patents and several more under development. You can find data from our technology in more than 50 peer-reviewed articles. We are proud to present the newest instrument, calScreener Plus. It is similar to the classical calScreener, but features wider temperature range from 20 to 45 degrees Celsius. In addition to traditional titanium vials, there will now be an option to run disposable glass vials with volumes up to 550 microliters. In both cases, 32 samples can be analyzed in parallel. calScreener Plus system consists of many different parts. The main unit is used to analyze the samples. The temperature control unit controls the temperature inside the machines. The classical calScreener was cooled down by the air surrounding it. calScreener Plus has an external thermostat that allows us to run the machine in lower temperatures. The whole system is controlled by the computer, provided by the customer, or can be purchased from us. A little bit also about biocalorimetry. Calorimetry is the science of measuring heat, the energy released from absorbed during any physical, chemical, or biological process. Microcalorimetry takes this concept to the microscopic scale, allowing us to detect extremely small amounts of heat, often in the range of microwatts. Biocalorimetry focuses specifically on living systems. Every cell in our body produces small amounts of heat as it metabolizes nutrients and performs work. By measuring this heat, we can monitor the cell activity in real time. For example, when bacteria grow and consume nutrients, they release heat. Microcalorimeter can detect this growth without needing any optical labels or dyes. Similarly, cancer cell treatment can be studied. When adding drugs to the cancer cells, we can observe drop in heat output that indicate that the drugs are working. Even simple processes like yeast fermentation or enzyme reactions can be tracked by heat that they produce. Other types of calorimeters can also be used for biocalorimetry, for example for study receptor binding and protein folding. In short, biocalorimetry gives us a window into the invisible energy flow of life, a way to listen to biology through heat. calScreener Plus is an isothermal microcalorimeter, meaning the measurements are done in a constant temperature. All metabolic processes produce heat. Biocalorimetry takes advantage of this principle. By measuring the tiny changes in heat flow, we can quantify the overall metabolic activity in a population of cells. The measurement reflects the free energy output of the entire sample, capturing both the intensity and dynamics of the cellular metabolism. Because the signal is heat, it integrates all ongoing biochemical reactions, respiration, growth, and stress responses, and gives you a continuous trace. This is possible thanks to extremely sensitive heat detectors, which can detect heat changes in the microwatt range. The underlying physics is rooted in the first law of thermodynamics, the principle of energy conservation. The energy cannot be created or destroyed, it only changes form. In a calorimetric system, which is essentially closed and thermally isolated, the biochemical energy converted by living cells appears as heat flow. Each sealed vial sits on a Peltier-based heat flow detector that is mounted on a heat sink. Released heat is transferred through the heat detector to the heat sink below and its surroundings. This temperature difference creates voltage that can be amplified and measured. The workflow for calScreener Plus is very simple and straightforward. Samples are directly loaded into titanium or glass vials that are placed onto a plate. The plate is loaded into the machines for continuous measurement. Since no dyes or labels are used, this method is non-destructive, meaning that the samples can be used for further analysis afterwards. Here is a short video of plate loading into the machine. The plate is placed onto the machine and pushed in with the loading arm. The loading is done in 2 steps, allowing temperature equilibrium. At the last step, the plate is slided onto the sensors. calScreener Plus comes with an intuitive software called calView. The software guides you through the plate loading and allows data monitoring in real time. As a result of a calScreener run, you can get a thermogram. Thermogram shows you a heat flow over time. Depending on the experimental question, different setups can be used to answer various questions. For example, the time that the thermogram appears is linked to the inoculum size and could be used to investigate the bacterial load. As the load decreases, the thermogram is shifted to the right. The peak height shows you the maximum metabolic activity and could be used to measure inhibitory effects or develop the best media for your bugs. So for example, if the media is better, the peak height is increased. There are many applications for this technology. In addition to previous applications in biofilm and other microbial research, this new machine opens doors for environmental microbiology. For example, studies of soil and water microbes that thrive at ambient temperatures 20 to 25 degrees Celsius. In addition, it's possible to study plant and algal biology, metabolic profiling of photosynthetic organisms. It enables yeast and fungal studies, characterization of eukaryotic microbes with optimal growth temperature below mammalian physiological temperature. It also opens the door for food and fermentation science, allowing the analysis of brewing yeast, probiotics, and food spoilage. We say that whatever fits in the cup, you can measure. 2 different sample handling kits are available for calScreener Plus, for titanium cal vials and for glass cal vials. The titanium comes with many benefits. Long-lasting titanium body minimizes the waste and reduces long-term consumable costs. Plastic inserts are available for easy cleaning and preventing of sample-to-sample contamination. It is compatible with a multi-channel pipette for 48 well plates. On the other hand, glass vials further minimize the risk of cross-contamination and are ideal for assays with precious material. They support experiments involving metabolic gas production and other analyses for downstream analysis. They also support workflows requiring sample injection. And the biggest benefit, the cleaning is extremely easy as they are fully disposable. If you have a hard time choosing the one for you, why not get both? The calScreener Plus system requires very little maintenance. The user will need to keep the system clean and tidy. This means making sure that no damage occurs to the connecting cables and removing any dust or contamination when it's present. The user will also need to restart and upgrade the computer as required by Windows. During the yearly visits, our service will take care of the rest. The service will calibrate the instrument and verify that the incubation temperature is correct. They will also perform any software or firmware upgrades available. To sum up, calScreener Plus enables real-time, labor-free metabolic measurement without dyes and reagents. Multi-channel design allows you to run up to 32 samples in parallel, making screening of various conditions possible. Increased temperature range expands studies to include environmental samples and organisms. Flexible sample formats allow you to work with opaque and complex matrices. Furthermore, non-destructive workflow enables post-experiment analysis, for example, microscopy, sequencing of qPCR. Thank you, and please reach out if you have any questions.